Welcome back to the deep dive. Usually when we tackle a subject like aging, the tone is, well, let's be honest, it's usually pretty somber.
It is. It's about decline.
Yeah, it's about things breaking down, joints creaking, that sort of inevitable slide. But the stack of research you've brought today feels different. It reads less like a medical eulogy and more like a software engineering manual.
It really does. I mean, it frames the whole problem through a lens that sounds like science fiction, but this is a very active, very heated medical debate happening right now.
And the core question is, what?
The question is, what if aging isn't about your body breaking? What if it's just about your body, forgetting how to work?
That distinction is huge.
Right.
If my car engine is broken, if the piston is shattered, I have to replace the hardware.
Right.
But if the car's computer just forgot the timing sequence, I could just reboot it.
Exactly. And that is the term the researchers are using, the factory reset.
Okay, so before we get into the mechanics of how you factory reset a human being,
Yeah.
we need to anchor ourselves. Today is Monday, January 19th, 2026. Why is this date significant?
It's significant because we are, in a way, crossing a finish line that was predicted years ago. We're looking at source material from Michael Ringel at Life Biosciences. And back around 2023, 2024, he flagged this year, 2026, as the pivot point.
The pivot to what?
He predicted this would be the year that the technology we're about to discuss moves out of mice and lace and into human clinical trials for reversing age-related diseases.
Now, this isn't just theory anymore. We aren't just talking about making super mice in a lab.
No. We are potentially watching this move into the clinic for real patients right now.
Okay, let's look at the stack. What do we have in front of us?
We have three big ones. First, a landmark 2023 study from Sel by Yang et al. This is the paper that basically rewrote the rules on why we age.
The why. Okay.
Then we have that 2025 presentation from Michael Ringel I mentioned. That gets into the how of reversing it, specifically using something called partial epigenetic reprogramming.
And the third piece.
A 2024 presentation from the Vadim Gladyshev. And this one throws a curveball. It complicates the picture by showing us that we don't just have one biological age. Your heart, your liver, your kidneys, they're all running on their own clocks.
So our mission today is to unpack this information theory of aging. We're going to figure out why scientists think there is a backup copy of our youth hidden inside our cells and how they're trying to hit restore.
But to get there, we have to start with the problem. The old theory.
The hardware view. For the longest time, my understanding, and I think most people's, was the wear and tear model. You go out in the sun, cosmic rays hit you, you breathe in pollution, your cells divide and make copy errors.
Right.
Basically, your DNA gets damaged, mutations pile up.
Right, like scratches on a car. Eventually, the engine just fails. That's the mutation theory. It's been the dominant dogma for decades. The idea is that the genome, the instruction manual itself, is physically degrading.
You just get ripped out.
Exactly. But the sources you brought, they suggest that's just wrong, or at least not the whole story.
It's not that mutation doesn't happen. It doesn't really explain aging. There were always these massive holes in that theory that scientists couldn't quite plug. Like what was the big contradiction?
The biggest one is cloning. Just think about Dolly the sheep.
Okay, the classic science class example.
But think about what it actually means. To make Dolly, scientists took a container of DNA from an adult sheep. That cell was old. It had lived out in the sun, it had divided, it had supposedly accumulated all those mutations.
So if the mutation theory holds up, that DNA should be trashed.
Precisely. If you put that old, damaged DNA into an egg and grow a new animal, that animal should come out old. It should have a short lifespan, maybe be born with arthritis.
But Dolly wasn't. She was born a lamb. She had a normal lifespan.
Which is a huge contradiction. It means the instructions for youth were still sitting there, completely intact inside that old cell. The manual wasn't torn up.
Hardware was fine.
The hardware is preserved. And this is where the metaphor shifts. If the genome is the hardware and it doesn't really change, then the problem has to be the software.
The epigenome.
Correct, the epigenome. If the genome is the piano, the epigenome is the pianist. Every cell in your body has the same piano, the same DNA. But the epigenome decides which song to play.
It uses chemical marks to tell a liver cell to play the liver song and a skin cell to play the skin song.
And under this new theory, aging isn't the piano breaking, it's the pianist getting confused.
That's a great way to put it. The Yang paper calls it epigenetic entropy. The cell loses its identity.
The pianist starts hitting wrong notes, a skin cell starts playing a few notes from the kidney concerto, it just becomes noisy.
And that noise is what we see as getting old. The cell forgets what it's supposed to be.
Yes. Now, the Yang paper didn't just theorize this, they proved it with this incredible experiment. They created a system they called ICE-inducible changes to the epigenome.
So how did they actually prove that software glitches cause aging?
They needed a way to scramble the software without breaking the hardware, so they found an enzyme from a slime mold.
Of course it's a slime mold. The unsung hero of biology.
They really are. This enzyme is called IPPI, and it's basically a pair of molecular scissors, but it's very picky. It only cuts DNA at non-coding sites.
The safe spots in the genome. So it cuts the DNA, but doesn't cause a mutation.
Exactly. It creates what's called a double-strand break, but no information is lost. The cell, however, doesn't know that. It senses a break and it panics. It sends out its repair crew.
And these repair proteins, they have a day job, right?
They do. Their day job is to sit on specific genes and regulate them, to keep the pianist playing the right song. But when the DNA gets cut, they have to abandon their post to go fix the break.
I see where this is going. It's like pulling the security guards off the front door to deal with a false alarm in the basement.
That's it exactly. They rush over, they fix the break perfectly, the DNA is repaired, but here's the glitch. When they're done, they don't always go back to the exact right spot. They get relocalized.
They get lost on the way back to their desk.
And if you do this enough times, cut, repair, get lost, cut, repair, get lost, the whole system gets noisy. The bookmarks are all in the wrong places. The cell starts to lose its identity.
So what happened to the mice? You cut their DNA, the repair crew got confused. Did the mice just, look a little tired?
Oh, it was drastic. Physically, they just fell apart. The paper details it vividly. These were young mice. But after a few weeks of this, they got gray hair, they lost their hair alopecia, they developed kyphosis, that hunchback you see in elderly animals.
Right.
Their bone density dropped, their vision failed. And just to be clear, this wasn't because of time. They were chronologically young.
But biologically?
When they measured their biological age with epigenetic clocks, these mice were aging about 50% faster than the control group. And I can't stress this enough, when they sequenced the DNA, there were no more mutations than in a normal mouse.
That is wild. The hardware was pristine, but the animal was dying of old age because the software was corrupted. Okay, this is terrifying, but it's also the hopeful part. Because if I scratch a CD, to use an old school analogy, the song is ruined. But if the CD is fine and the laser's just skipping, I can fix the laser. I can reboot.
And that leads to the trillion-dollar question, can we do it? Can we polish the scratch?
Which brings us to the factory reset and Michael Ringel's work, the concept of the backup copy.
This is the part that just blows my mind. The theory suggests that inside every single old, decrepit cell, there is a backup copy of the original epigenetic information, a pristine version from when you were young.
How do we know that? Is that just wishful thinking?
We know it because biology does it every single day. Ringel used this really poignant example in his presentation. He showed a picture of his wife and their new baby.
Okay.
Now think about the biology. His wife is, say, in her 30s. Her egg cells are biologically 30-something years old. The father's sperm is also old in cellular terms.
Two old cells meet?
Right. But the baby isn't born 30 years old. The baby is born at biological age 0, ground zero.
I've never really stopped to think about how weird that is. If damage just accumulated linearly, every generation would be born older than the last. We'd be extinct.
Exactly. So somewhere in there has to be a massive clearance event, a reset. And Ringel explains that this happens between day 7 and 9 of embryogenesis. The embryo just wipes the chalkboard clean.
It restores the software to version 1.0. So the goal of this whole field is to trick an adult cell into hitting that reset button.
But, and this is a very important safety constraint, without turning the whole person back into an embryo.
Right. If you turn a liver cell all the way back, you don't have a liver anymore. You have a pile of stem cells, which is basically a tumor.
A teratoma. Not ideal.
Yeah.
So the control is everything. This is where the Yamanaka factors come in.
Okay, OKSM.
Right. Shinya Yamanaka identified 4 proteins that can do this. OCT4, SOX2, KLF4, and COSKM.
But there's a catch with the M.
A big one. c-Myc is an oncogene. It drives cell growth, but it pushes the pedal to the floor. It causes cancer. So the breakthrough Ringel talks about is actually about restraint. They drop the M. They just use OSK.
And that works. It doesn't just do 3/4 of the job.
It seems to be the Goldilocks solution. It pushes the cell back in time just enough to restore its function and identity, but not so far that it becomes a stem cell.
Okay, theory is great. Let's talk results. What did they actually achieve with OSK?
The data Ringel presented is just stunning, specifically because they targeted the eye, which is part of the central nervous system, very hard to regenerate. They looked at glaucoma and a condition called NAION. It's like a stroke of the optic nerve. In the mice with glaucoma, the OSK therapy restored their vision, not just stopped it, reversed it. In the stroke model, they saw a complete recovery of vision all the way back to baseline.
But mice aren't people. Did they test this in primates? That's the real hurdle.
That's where most drugs die. But yes, Ringel showed data from non-human primates. Now, obviously, you can't ask a monkey to read an eye chart, so they measured the electrical signals in the brain.
The signal from the eye to the brain.
Correct. After they damaged the optic nerve, that signal was flatline, obliterated. But after the OSK treatment, the signal came back. It looked almost identical to the pre-damaged signal.
That's incredible. We're talking about regenerating the central nervous system in a primate.
And crucially, the safety data showed no tumors, no adverse effects.
Which brings us back to today, January 2026. This is why he circled the date.
Exactly. He said that phase 1 human trials for these conditions were expected to start right about now. We are no longer asking if this is possible. We are asking, does it work in humans?
It's fascinating to think we might be treating glaucoma not by lowering eye pressure, which is just managing a symptom, but by literally making the eye younger.
It completely changes the definition of the disease. But you knew there was a but coming.
There's always a complication. It can't just be take a pill and live forever.
Well, we've been talking about aging like it's one single dial. But Vadim Gladishev's research, it throws a wrench in that simple picture.
This is the organ clocks idea.
Correct. We don't age all at once. Gladishev's team found that you might be 45 chronologically, but you have a liver age, a heart age, a kidney age, and they are rarely the same.
So I could be 40, but my liver is 55.
Exactly. In fact, he found that about 2% of people have specific organs that are aging much, much faster than the rest of their body.
That is unnerving.
But it's also actionable. It explains why some people get heart disease first or kidney disease. Their organ clock is just running faster there.
Gladyshev also pinpointed when this all starts, and this was the fact that just blew my mind.
The ground zero moment. He found that aging essentially begins in the womb. Remember that reset we talked about, day 7 to 9?
Right, the software wipe.
Well, immediately after that, as the embryo starts to develop, the clocks start ticking. You are technically aging before you were even born.
The minute the reset is done, entropy starts creeping back in.
It does. And he describes it as a non-autonomous process. The clocks influence each other. If your heart ages, it stresses your kidneys, which then makes the kidneys age faster. It's a cascade.
You had some really specific, almost quirky data on lifestyle factors. I have to ask about the alcohol and yogurt thing.
The proteomic data, yes. Okay, big disclaimer here. These are associations from a huge data set, not medical advice, but the correlations were fascinating. Smoking, obviously, ages the lungs.
No surprise there.
But it had less of a negative impact on the skin and arteries than you might expect. Not zero, just less. It's organ-specific damage.
And the white wine.
This is where it gets so nuanced. Alcohol generally sped up aging in the kidney and the intestine, but white wine specifically showed a slight beneficial association with arterial aging.
So, good for the heart, bad for the gut.
That seems to be the trade-off in the data. But then he noted that yogurt was associated with reduced intestinal aging.
So if I have a glass of Chardonnay, I should chase it with a yogurt.
Laughs. I'm definitely not signing off on that cocktail. But the bigger point is that healthy isn't a generic term, it's personal.
And that leads to the future of this. It's not about living forever. It's about targeted maintenance.
Personalized longevity, exactly. If a clock says your liver is aging too fast, maybe you use a targeted OSK therapy just for the liver. Ringel actually mentioned they have data showing OSK works in the liver too.
So we have the science, we have the potential treatment, but then there's a question of should we? And surprisingly, a lot of people say no.
The moral imperative. Ringel brought this up, setting a Pew study. It found that 51% of people thought medical treatments that slow the aging process were actually bad for society.
Bad for society. That seems so counterintuitive. Who wakes up and says, I really hope I get frail soon?
I think people hear slowing aging and they immediately think immortality for billionaires. They worry about overpopulation, resource depletion, that it's unnatural.
But Ringel pushes back on this pretty hard.
He reframes it completely. And I think this is the argument that really cuts through the noise. He asks, if you have a patient with glaucoma and you can restore their vision by reversing the age of the cells, is that anti-aging or is that just medicine?
In medicine, you're curing blindness.
Exactly. His argument is that aging is the driver of these diseases. If you treat the age of the cell, you treat the disease. To refuse to use these tools because they involve aging is to refuse to cure blindness.
It's a powerful argument.
Yeah.
We don't really have moral debates about curing cancer or heart disease. But because this mechanism is rejuvenation, it just feels different.
It feels different because it challenges our fundamental acceptance of decline. We've accepted that getting old means breaking down. They're arguing that decline is a treatable condition.
So let's bring this all together. What does this mean for us right now in 2026?
I think we are witnessing a paradigm shift. We have moved from thinking the car is broken, the hardware mutation theory, to realizing the driver is just lost. That's the epigenetic software theory.
And we found the map.
We found the map, the OSK factors. And thanks to people like Gladyshev, we are learning to navigate the specific routes, the individual organ clocks. We aren't just blasting the whole body, we're learning to be precise.
It really changes how you view your own body, that you aren't just one static age, you're a collection of systems, some running fast, some slow, all managed by software that we finally figured out how to reboot.
And that raises a provocative question for me. If chronic diseases like heart failure or kidney disease or glaucoma, if they're really just pockets of accelerated aging in specific organs, does the word disease even make sense anymore?
That is a deep thought. Is it a disease or is it just local clock running too fast?
And if we all have this backup copy of youth inside us, this pristine software, how many times can we hit restore? Is it a one-time fix, or could we theoretically keep rebooting the system indefinitely?
A backup copy of youth. It's a comforting thought. And who knows, maybe by the end of 2026, we'll have the first human data to prove it actually works.
We will be watching.
Thanks for diving deep with us.